Good morning, dear students. Today we are going to study about bioremediation of radioactive waste by microbes. Radioactive waste bioremediation. Bioremediation has been developed as a potential approach that can be implemented in radioactive waste remediation. Bioremediation stabilizes and reclaims radionuclides and toxic metals from contaminated materials, soil, sediments, or waste. Bioremediation process involve the metabolic activity of microorganism in situ. It involves relatively low cost, low technology techniques and have a high level of public acceptance. So, radioactive bioremediation. Bioremediation is evolved as a new branch of environmental microbiology, which is mainly dealing with handling environmental waste degradation, and it is implemented to biodegrade waste like xenobiotics as well as radioactive waste. And bioremediation, it helps to stabilize and reclaims this radioactive waste and toxic metals associated with this radioactive waste, and it helps to eliminate such type of waste materials from soil, sediments and other waste products. And bioremediation, it involves usage of microorganism either at the site of production or at the ex situ site, that is a site away from the origin of this waste. But in case of radioactive waste, since it is highly hazardous waste, most of the bioremediation activity are taking place at the site of production itself. So it is here the activity of the microorganisms are in situ, that is at the site of production of the waste itself, microorganisms are applied to degrade this waste, and it involves relatively low cost and low technology needs, and it is having a high level of public acceptance. Sources of radioactive waste. The sources of radioactive waste include testing of nuclear weapons, nuclear energy generation programs, nuclear weapon reprocessing and accidental release. So these are the sources from which the radioactive waste get into the environment. So while we are testing the nuclear weapons and where the nuclear energy is being generated, and when the nuclear weapon is reprocessed, and the accidental release of these radiations or the radioactive elements from the nuclear reactors. So these are the different sources of radioactive waste. Microbial bioremediation. Mainly bacteria is involved in this bioremediation of the radioactive waste. Fungus is also employed, but to a lesser extent. Then when we are using fungus for bioremediation, then it is called mycobioremediation. Then anaerobic bacteria are used to concentrate, contain and stabilize the toxic metals and radionuclides in the waste. Bacterial genera such as Geobacter, Deinococcus, Shivanella, Serratia, Echinococcus radiotolerans and Hymenobacter metalli are used, which can act on radionuclides present in a high level of radioactive waste, affecting their solubility, mobility and bioavailability. So different types of bacteria can be employed for this bioremediation of radionuclides. Anaerobic bacteria are commonly used to concentrate and stabilize toxic metals as well as radionuclides, and bacteria of the genera Geobacter, Deinococcus, Shivanella, Serratia, Echinococcus radiotolerans and Hymenobacter metalli. These are some of the commonly used bacteria which can act on these radionuclides present in this radioactive waste, and it affect or it eliminate this radioactive waste by in interfering with their solubility, mobility and bioavailability. Then microbial mechanisms involved in bioremediation. The successful bioremediation of radionuclides relies on complex interactions of biological, chemical and physical process. So mechanisms include oxidation, reduction, dissolution, precipitation, absorption and leaching. So through any of these mechanisms or through a combination of one or two mechanisms, these microorganisms are biodegrading the radionuclides. So the biodegradation of radionuclides by microorganism is a complex process which involves the interaction of biological, chemical and physical process, and these are the various processes through which the microorganism degrade these radionuclides. Role of Bacillus sphaericus to trap uranium ions. Role of Bacillus sphaericus to trap uranium ions. So this is actually a work done by some of the eminent scientists abroad, and they were working with this Bacillus species, and they have found out that this Bacillus species is capable of absorbing uranium or filtering uranium, and it is been used in large scale to remove these radionuclides, and it is also used as a extraction method for extracting uranium for reuse in power plants. So this Bacillus sphaericus, it is a microbe that is a bacteria which can bind and accumulate high amount of radioactive metal. So this Bacillus sphaericus, it has the capacity to accumulate high amount of radioactive metals, especially uranium. So it can thrive well in this uranium mining waste piles, a toxic environment which contain other metals including copper, lead, aluminum and cadmium along with uranium. So these are all heavy metals or are heavily dangerous metals along with uranium, which is a radionuclide. Other metals like heavy metals like copper, lead, aluminum and cadmium present environment, this Bacillus sphaericus can thrive well, and it was reported to possess a paracrystalline proteinaceous surface defined as S layer, to which these toxic metals are found to localize. So this bacteria is having a S layer, and to this S layer, this is a proteinaceous surface, a paracrystalline proteinaceous surface within the bacteria, which is able to localize or concentrate these heavy metals. I mean these heavy metals as well as this uranium, especially uranium nucleoids. A filter design utilizing this bacterial S layer to trap uranium ions as a wastewater or sediments are passed through the filter. So this S layer of this bacteria is used for preparing a filter or used to design a filter, and this bacterial S layer, it is capable of filtering all the uranium ions as the waste or sediments containing this uranium is passed through this filter. In immobilizing the radioactive uranium species on the S layer, it becomes much easier to remove large amount of radionuclide. So if we are able to fix this S layer with the bacteria, or we have to immobilize it at a particular region for a long period, we can extract a large amount of uranium residues. Through further chemical treatment, it may be even possible to recover some of the waste uranium for reuse in power plants. So, by further chemical treatment, we can reuse some of the uranium that is trapped by the S layer of this bacteria for reuse in power plants. So, this is about the role of Bacillus sphaericus to trap uranium ions. So, Bacillus sphaericus, it's a bacterial species which is able to biodegrade or which is able to trap uranium that is present as a major radionuclide waste in the waste along with the other metal species. Next is mechanism involved in microbial bioremediation. The microbial bioremediation of radionuclides is of paramount importance to the development of new strategies and techniques to protect the environment. Various mechanisms involved in microbial bioremediation of radionuclides are. So there are various mechanisms involved in this microbial bioremediation. The first one is biopsion and bioaccumulation. So first mechanism involved in this microbial bioremediation is biopsion and bioaccumulation. Positively charged metals and radionuclides are attracted towards negatively charged surface receptors and cell organelles of bacteria. So if the radioactive waste contain positively charged radionuclides as well as positively charged metals, they are attracted towards the negatively charged surface receptors and cell organelles of bacteria. This extracted radionuclides are precipitated by slime and capsule produced by the microorganism. Thus we can extract that radionuclide waste. Okay, so by biopsion and bioaccumulation, positively charged radionuclides can be attracted towards the negatively charged receptors as well as organelles of the bacteria, and this bacteria help to bioaccumulate such radionuclides in their slime and capsule. So this is the first mechanism. Second mechanism is direct reduction of radioactive heavy metals by microorganism. So microorganism directly reduce these radioactive heavy metals. Soluble forms of radionuclides are converted into insoluble precipitate by the direct reduction of radionuclides by the action of enzymes present in microorganisms such as reductase and hydrogenase. So it is an enzymatic reduction. So here enzymatic reduction of radioactive heavy metals is taking place, and here the enzyme is produced by the microorganism, and the enzyme involved are reductases and hydrogenases. So these enzymes present in the microorganism form insoluble precipitate of the radionuclide. So actually if the radionuclides are so insoluble form, then they will create much problem, because they will easily get solubilized in our drinking water, or they will easily leach out to our groundwater, and thus they reach our drinking water services, and they can create a lot of health hazards. But this microorganism, through their enzymatic reduction, they can convert these soluble radionuclides to insoluble precipitate, and thus we can eliminate such type of heavy metals or radionuclides. Next method is indirect electron transfer by metal reducing microorganisms to radionuclides. So indirect electron transfer by metal reducing microorganisms. So bacteria reduce the metals available in the surroundings. The byproducts formed in this process help in the chemical reduction of radionuclides. So in most cases the radionuclides are seen associated with a lot of heavy metals. So first bacteria reduce the metals available in the radioactive waste, and the byproducts formed by the reduction of this metal help in the chemical reduction of this radionuclide. So at first bacteria reduce the metals, then the products that is produced by the reduction of the metals help in the chemical reduction of the radionuclides. So radionuclides get converted into less toxic elements. Then next mechanism is controlling the solubility of the radionuclides. So microorganisms have the potential to control the solubility of the radionuclides either directly through interaction with the microbial cell or by indirect transformation driven by local changes in the environment attributed to the microbial activity. So microorganisms have the ability to control or change the solubility of the radionuclide either by directly interacting the radionuclide with the microbial cell or by changing the environment in which the microbial activity is taking place or this radionuclide is present. So by changing that environment, the radionuclide become insoluble or it cannot solubilize in that environment. Next is biomineralization of the radionuclides. Radionuclides. Then chelating agents which are used to precipitate nuclear waste are naturally formed by some bacteria. So in biomineralization, the chelating agents that are used to precipitate nuclear waste, they are generally formed by some bacteria. For example, this citrobacter, it produces citric acid which acts as a chelating agent in decontamination of nuclear waste. So here bacteria will produce chelating agent which will precipitate nuclear waste so that their toxicity will be reduced. So an example is citrobacter. It will produce citric acid. It is a chelating agent and it will cause the precipitation of these radionuclides and thus it is decontaminated. Next is biostimulation. So we can in biostimulation we are using diverse microbes having synergistic action to areas highly contaminated with heavy metals and radionuclides to speed up the process of bioremediation. So in biostimulation we are using so many groups of microorganisms to a particular area. We are introducing diverse amount of microorganisms to a particular area highly contaminated with radionuclides, and this introduction of these diverse microbes will speed up the process of bioremediation. So this application of microorganism to a particular area will speed up the bioremediation process. Next is production of GMOs. Production of GMOs means we have to genetically modify the genome of certain organisms, certain particular group of microorganisms, so that through genetic engineering, so that they can be considered as a genetically modified organism and they can be applied to biodegrade this type of radioactive waste. Bioremediation by Deinococcus radiodurans. So Deinococcus radiodurans, it is one of the most radiant resistant organism. It does not possess the ability to degrade toxic compounds that are leached from radioactive site. Even though it can eliminate or it can convert uranium ions, it is not able to degrade all the toxic compounds that are leached from the radioactive site. Genetically engineered variants of this Deinococcus radiodurans can perform this function of degradation of toxic compounds other than uranium from this radioactive site. So degradative genes from Pseudomonas could be easily introduced into this Deinococcus radiodurans, which will make it more robust in bioremediation. So we can incorporate genes from Pseudomonas into this Deinococcus for making it much more good in degradation of toxic compounds. So that this is done by genetic engineering, and genetic engineering create recombinant strains having supreme capacity of degradation of the environmental pollutants. For example, insertion of mercury resistant gene which is called MER-A from E. coli into Deinococcus radiodurans, making it capable for withstanding 60 GV per hour of radiation, a dosage that found in a dosage higher than that found in most radioactive waste sites. So this mercury resistant gene MERK, it is isolated from E. coli and it is been genetically incorporated into radiodurans, and making that Deinococcus radiodurans able to withstand 60 GV per hour radiation. That is a dosage which is very higher than that of that found in radioactive waste sites. So Deinococcus radiodurans can be genetically modified by the insertion of mercury resistant gene from E. coli. Then higher radioactive resistance correlate to an ability to convert toxic volatile metal species into less reactive, less toxic form. So this higher radioactive resistance. So this Deinococcus radiodurans is now having a high radioactive resistance. That means it has the ability to convert toxic volatile metal species into less toxic and less reactive form. Introduction of this MERK, a gene coding for mercuric ion reductase, into Deinococcus radiodurans, was found that the organism was capable of converting the toxic mercury ions to elemental mercury. So with the incorporation of this particular gene for the mercuric ion reductase, this Deinococcus radiodurans was able to convert toxic heavy metal non-metal mercury ions into elemental mercury. Multiple remediation strategies have been introduced to a single strain of organism through insertion of various gene clusters. So, to a single organism, we can incorporate genes from different group of microorganisms so that it can be used for the bioremediation of various types of radioactive waste. Introduction of other genes for toluene and chlorobenzene metabolism to Deinococcus radiodurans make it possible to use a single strain of bacteria to clean up multiple contaminants associated with the nuclear waste site. So, introduction of other genes from other group of bacteria help the Deinococcus radiodurans to metabolize toluene and chlorobenzene, which is seen associated with this nuclear waste. So this is all for today's topic, and these are the references from which I have taken this note. As it is a relatively new topic, I have taken notes from the test, and these are the first two are the important works made by some scientists. So the Bacillus pyrrhicus, it is been the metal binding bacteria from uranium mining waste piles in technological application. The original work was done by Polman et al. And the next work regarding this Deinococcus radiodurans was done by H. Brim et al. And these are other sites from which I have taken these notes. So thank you.